This video is going to focus on DNA replication, or how DNA makes copies of itself. We've already talked about why we need copies of DNA, but now we're going to look at how that actually happens and the steps and different enzymes that are involved. Here we have a nice visual showing the cell cycle. So you will remember that when the cell is first in G1, it's growing, it's still metabolically active, but in each cell there's only one copy of each of the chromosomes. So in human cells we have 46 chromosomes. Each one consists of a single chromatid. But at the end of the S phase, that's when DNA replication occurs. I think about the S as synthesis. So at the end of the S phase, each chromosome is now made up of two identical sister chromatids. So this is the stage we're going to focus on, the S stage, because that's when DNA replication occurs. And remember, the reason that the DNA has to be replicated or copied is because after we go through the rest of interphase and enter mitosis, the chromosomes are all organized and arranged so that the identical sister chromatids are separated. So when you have two cells, each one has its own copy of the DNA. So DNA replication occurs during the S stage of the cell cycle, of interphase, and that's in order to make copies of the DNA in preparation for division. Now we'll talk about how DNA replication works. We can see here we have a standard ladder model of DNA, and remember that DNA, the building blocks, are these things right here. So this is one nucleotide, and each nucleotide consists of a 5-carbon sugar, in this case deoxyribose, and a phosphate, and then a nitrogen base. Remember the sugar-phosphate backbones, and then the nitrogen bases make up the rungs of the ladder, and they're held together with hydrogen bonds. So that's what these dotted lines represent. Also, you will recall that the nitrogen base pairs are always complementary. So whenever we find A, or adenine, we find T, thymine, on the opposite strand. G for guanine, C for cytosine. Now, you may have thought about this and gone, since due to that complementary base pairing, maybe if I unzip that double helix, now each strand could act as a template to build a new strand. And that's exactly what happens. And there are a couple of important enzymes involved in this, so let's look at those and what they do now. There's a lot going on in this picture, but we're going to focus on a few things. First, we have a chromosome, and if we zoom into that chromosome, we can see our DNA double helix. Now, this light blue right here represents the sugar-phosphate backbone. That's the original, or the template strand. So that's the one that's being copied or replicated. The first enzyme that we want to focus on is this one right here, helicase. Okay. So helicase, I think kind of sounds like helix, right? Helicase, helix, and it has the ASE enzyme ending. And what this does is it unzips the double helix. And what that means is that it's actually breaking the hydrogen bonds that are holding the nitrogen bases together. And it breaks those hydrogen bonds, separating the two complementary strands from one another. The second enzyme that we are concerned with is this one right here, DNA polymerase. And you can see it acting on both sides of the strand. And think DNA polymerase, it has polymer. And still has that ACE ending, right? Polymer. So if you remember, polymer is made up of lots of units, lots of repeating subunits. So DNA polymerase is making that larger DNA molecule by adding nucleotides to the new strand. So if we look at what's happening in this picture, the helicase is unzipping, breaking apart, and then we're left with these two light blue template strands. And then the DNA polymerase comes along, and it reads the nitrogen base. So it says, all right, here we have, what's down here? This yellow one is a thymine, so we're going to add an adenine onto it. And then we have a guanine, so this cytosine, we're going to pull that in, and so on and so on. So it's able to read the template strand and build the new strand. So these darker blue strands right here are the new strands of DNA that are forming. Here's a simpler model showing what's happening. So we have our complementary base pairing. Complementary base pairs. Right, so remember, A with T, C with G. So that's going on. We can see that here in Model 1, or A, I should say. Part B That unzipping or breaking of the hydrogen bonds between the nitrogen bases is done by helicase. And then part C is when the DNA polymerase reads the exposed nitrogen base and adds on the complementary base pair. And then in the end, you end up with two strands. Notice they are identical, right? So these two identical strands in chromosomes would make up the two identical sister chromatids. DNA replication is oftentimes described as semiconservative replication. I think semi means half, and then conserve, right? Save or keep. So what does that mean, semiconservative replication? In terms of DNA, here's our old strand represented with purple, right? Acts as a template to build the new strand. And if you look at the two new double helices, each one is half old, half new. So that's what semi-conservative replication means. And that helps to ensure that replication is consistent and there are fewer mistakes because each original strand acts as a template for the new one, and as long as those complementary base pairs are always matched up together, we should be able to replicate this a bazillion times without any mistakes. Doesn't mean mistakes don't happen, but it is a pretty good system of checks and balances.